Today what I'm going to do is give a little bit more of a research-focused talk. I'll talk a bit about some experiments done by other people and some experiments done in my lab. So just to sort of briefly summarize where we were yesterday, I gave just a brief introduction in the last 20-25 minutes of yesterday's lecture about genome dynamics and experimental evolution. And particularly, we talked about how these experiments were set up, and we talked about this first case of looking in detail at the dynamics of genome evolution in an experiment from Jeff Barrick's paper on the evolution of one of Rich Lenski's long-term lines through 20,000 or so generations of evolution. And we, talked about these mutations that they found and the degree of repeatability. And then where we left off was we were talking about how we would like to ideally be able to really understand how the sort of things that you get in these kinds of experiments, the results that you get, depend on the sort of spectrum of things that could happen via all these dynamical processes that I talked about in the first two lectures, how mutation, selection, and drift interact in rapidly evolving populations to pick and choose from among these possibilities to give you what you actually see. And hopefully, at least in some sort of rough sense, we could try to understand why we get the mutations that we get in these experiments and why we see the degree of repeatability that we see across replicate lines. And I left you with this sort of idea that ideally what we would like to do is address these kinds of inherently statistical questions about the probabilities that different sorts of mutations fix in replicate lines. We would be able to ideally address them by looking at lots and lots of evolution experiments in parallel. And so this is a liquid handling robot that we use in my lab to these are microplates. Each of them has either 96 or 384 of these little tiny test tubes in them, effectively. And so, on one of these decks, we can maintain thousands of populations in parallel and just sort of pipette things around with the robot, and then we can measure fitness with these high throughput flow cytometers. Now, the thing, though, that was difficult until very, very recently in these kinds of evolution experiments is that although you can maintain thousands of lines in parallel, and you can measure phenotypic characteristics of thousands of lines in parallel, if we want to look at the genome dynamics of thousands of lines in parallel, although the sequencing capacity itself is not particularly limiting, we can sequence in terms of just the pure cost of the sequencer for about $10 a strain to quite good coverage. The library preparation has been very expensive. So it depends on exactly how you do it. But until recently, it would cost you at least $50 per library, meaning that, and more like $100. And if you do it, if you send it off, maybe $400 or $500 per library. And what this means is that you can, how do I want to put this? For every sort of independent strain that you want to sequence, you have to prepare the sequencing library to send to the sequencing machine. And so it would cost you, this much to prepare that sequencing library. And so that would mean that you couldn't really take 1,000 independent clones and sequence them independently. Okay, what you could do, however, is just mush up the whole culture in one big mush and sequence that whole thing as a single library. Okay? And so you couldn't sort of identify from that the genotypes of individual strains, right? But you could see something about all the variation that was present in the population as a whole. And so what I'm going to tell you about to begin with is a couple of experiments, one of which actually did sequence individual clones, but was limited to of order 100 just because of these costs. That's $10,000 to prepare those libraries. Or a little bit later, I'll tell you about an experiment in my lab which uses this sort of mush everything up approach. Now, a postdoc in my lab has managed to get this down to under $5 per library, and so now we're sort of launching experiments where this is not a limitation, but those are not done yet. So here's the first experiment that I want to talk about. There aren't really that many experiments of this type that have been done, so I can sort of tell you about more or less all of them. So this was a study done by Olivier Tenelion when he was on sabbatical in UC Irvine in Tony Long's group, and with Brandon Gout as well, I think. And he wanted to look at the spectrum of evolutionary responses to heat stress in E. coli. So he took 115, I think it was, populations of E. coli and evolved them to, I believe it was 42 degrees or something like, was it 42? It was high temperature anyway. I think it was 42 degrees for 2,000 generations. Okay? And he took a single clone from each of these adapted lines, okay, limited to 100 by these library preparation costs. And he sequenced each of these clones. And what I'm showing you here is a figure from this paper that he wrote about this work, where each of these sort of little lines represents one of the genomes, one of the E. coli genomes that he sequenced. And each of these little colored marks represents some mutation that he found. So these red mutations are just, point mutations, single nucleotide polymorphisms. Okay, these yellow things are indels. These purple things are larger deletions, and there are various other types of mutations. And I kind of like to look at this. I mean, to me, this is just wild. Like, they told me about they were doing this experiment before they got the data, and I just thought, man, you're just not going to see the same thing twice. Because, I mean, 100 lines, there's genome of several megabases. What's the probability that you get the same mutation coming up in multiple lines? But man, I was definitely wrong. And you look at this, I mean, look at this position here. Something like 2/3, more than 2/3 of the population's got a mutation at this position. Many of these lines got this large deletion here. But then you also see a lot of stuff that's only present in 1 or 2 replicate lines, maybe a few replicate lines. And you can kind of get a sense for this. This is sort of scanning along the slightly less than 5 megabase genome here and looking at the frequency at which you have a mutation as a function of position. So you see some positions which really get mutated again and again in replicate lines and others that don't. And so the question is, why the hell do you see this, right? What is driving this particular pattern of repeatability that you see? And why isn't there more? Why isn't there less? Why are these particular sites involved? These are the kind of questions we'd like to address. And I mean, I should just say up front, disclaimer, we're not going to answer any of these kinds of questions today. We don't know. This ability to do this kind of stuff is very new. That first paper was 2009, and as you'll see, all this work is just over the past couple of years. So this is just beginning, but I'm going to hopefully sort of set the stage of the types of questions we can at least start to try to address. Okay, yeah. Sorry? It's large. I don't remember exactly the details, but it's large, like 10 to the 5 at least. I think it was larger than that, like 10 to the 6 at least. I mean, it was just E. coli in test tubes, so whatever. I don't remember exactly. Do you remember, Deepa? Something like that, yeah. But they only did one population size. And yeah, another sort of interesting question you can ask about this is, could you look at this data and say, if I were to reduce the population size or increase the population size, what would I get? I mean, presumably, if I increase the population size, it ought to become maybe more repeatable, would be my sort of rough guess. This mutation would occur in a larger fraction of the lines, let's say. But I don't know. Would there also be more one-off mutations? Probably that would be true as well. Okay. So sort of remarkable that these guys seem to saturate the total number of mutations, I mean, depending on how you look at it. So this is just a rarefaction curve showing the number of sequence lines up to about, I think it was 115. And these are the sort of total number of unique mutational events that they saw. And this light blue line is the one I want to focus on, which is the sort of total number of different genes in which they found at least one mutation, right? So you can see that, they, after doing even after doing 20 or 30 lines, basically every gene that was going to be mutated across many, many replicate lines, they had seen at least once, right? And so, you're really sort of exploring at least all the genes that you're going to see mutations in these lines over this sort of scale. So there's really a tremendous amount of parallel evolution across these replicate lines. This red line is just at the point mutation level, and this is just all mutations. But I mean, even at point mutations, you're seeing exactly the same nucleotide change in replicate lines across replicate populations. So, they're seeing exactly the same base pair in the genome mutated, just completely independently in different lines. Just to give you a sense, what are some of these mutations? So here's just a bunch of examples of some of the most commonly hit mutations. And many of them are things that sort of make sense as being important for adaptation to heat stress. Others of them were not. And you can see some of these, like, for example, RNA polymerase. In 114 lines, there were 205 mutations, because, I mean, this is clearly something that you really want to change in some way in response to this heat stress, so much so that many lines got mutations in multiple of these different genes. So one really interesting conclusion of this particular study was that, to some extent, epistasis drives the degree of repeatability across replicate lines that they see. Or at least it describes some of the patterns in the data. So all of the sort of theory that I told you about over the past couple of days has been assuming sort of simple models of directional selection, where you just have some distribution of fitness effects, which doesn't change as the population adapts. And this figure is evidence that that does not hold, okay? At least in some ways it does not hold in these experiments, and that fact is responsible for at least some aspects of the data. What we're seeing here is Olivier manually grouped all the mutations into various functional categories described by here. So these are like RPO, ABC. They're various different sort of functional categories of the mutations that he observed. And then each of these sort of columns and rows here is a single evolved clone, okay? And so what he did is he plotted, for example, this evolved clone. If it shares, sorry, put it the wrong way. Each of these lines is a single evolved clone that got a mutation in this particular location, in this particular path, in this particular function, right? So, for example, this point represents one evolved clone that got a mutation in this particular group, and then all the other mutations that clone got are listed over here, okay, right? And so what you can see, and the same thing is sort of up in the column, and so what you can see here is that there's really a big sort of reduction in the number of points along the diagonal, okay, which suggests that basically each of these functional categories, maybe with the exception of this one, but each of these functional categories, you get one mutation, and that's enough, right? And there may be many different genes in these different functional categories, but you just get one, and that's it. And so this suggests that maybe there are 1, 2, 3, whatever, however many this is, a dozen different mutations, right, that you can get, and then you're adapted to this heat stress, right? Like you can get, one mutation in each of these categories, but getting 2 in one of the categories doesn't do you any good, right? And so then maybe, there are sort of sets of equivalent mutations, but there are only a sort of small number of such sets, and you get all of them, and then then you're done, right? And so the degree of repeatability is just, okay, 2 lines are going to be more similar if they get the same mutation within this set, and more different if they get different mutations within this set. And some of these sets are small and have lots of mutations. Some of them are a little bit rarer, so on. You also see some sort of, yeah. Yeah. Yeah, I mean, this is just, presumably there is some neutral stuff going on. But all of this stuff, I mean, this also suggests that almost everything is is adaptive as well, right? So yeah, it seems like at least the big majority of what you get is adaptive. And I forget what DNDS is, for example, in these experiments, but it's enormous. In Rich Lenski's experiment that I told you about, it was actually infinity. There were... Well, it's, yeah, I mean, at least with the mutation rate in E. coli. I'll show you about yeast experiments over similar timescales where you get lots of neutral mutations fixing. But yeah. No. Would be interesting to do, right? Olivier has talked about doing that, but right, yeah, there are old experiments like that, but no sequencing data. I mean, there's a really cool experiment in flies that does that. Teutonio, I think. There's a paper in Nature Genetics, but that's very different because you're looking at selection. You know, you're starting with outcrossed lines, and so it's like selection and standing variation. Then when you reverse, like nothing is fixed, and so you, yeah. Yeah.
So I'm curious about these really dark lines that go, for example, the del1.
Yeah.
So what's going on there? So if you have the del1 mutation, you're more likely to be mutated in other mutations.
I guess what this means is that every line has a mutation in del1, basically. If I'm understanding that correctly. All right. You can kind of look at this in various ways. So they sort of looked at positive and negative associations between these groups, and they saw, you know, apart from the obvious diagonal, there were some sort of larger scale groups where you tended either to be a little bit more or less likely than chance to get mutations, you know, in multiple of these functional groups. I don't know. I kind of think it's a little bit easier just to look at this, and you can take that for what it's worth. All right, but so this experiment I thought was really cool, and somehow it's a signature of what's going on here, right? We're seeing some set of outcomes with some parallelism, and you know, it's a function of the details of the dynamics in their experiment and the sort of, you know, set of things, all these different functional categories and how many mutations can happen in each and what their fitness effects are. And so one of the things that I wanted to do in my lab following on from this is to look at the actual dynamics directly that is sort of picking and choosing among these possibilities. So this experiment just looked at the final time point, right? After 2,000 generations, they sequenced a bunch of clones. So we don't know how this sort of progressed over time. And so what I wanted to look at was this dynamics process itself and sort of see if we could say anything more about, you know, which mutations are sort of trying to fix and which ones actually do, okay? And as I mentioned last time, right, the basic difficulty in making this mapping is that, you know, in the small populations, this is the sort of regime where mutations are rare. I'm going to show these sort of Muller diagrams in interpreting the data, so I just want to sort of recast what I talked about in the past couple of days in those terms. You know, when mutations are rare, this is a sort of Muller diagram like I drew once or twice, where, you know, you get some beneficial mutation, it sweeps, and another one later, and so on. And in this regime, it's sort of very straightforward to figure out which mutations are going to fix and which aren't, right? It's just mutations occur according to their mutation rates. They fix if they survive drift. And so the distribution of fixed mutations, the mutations that fix, is a sort of straightforward function of the mutations that can occur. But when you've got lots of clonal interference and multiple mutations, you've got a sort of whole swarm of things going on. And this is sort of what I talked about the past few days. And you know, now we have some theory to describe how we get from the distribution of things that can happen to which mutations actually survive this process and fix. But we wanted to just look directly at this dynamics. Okay. So this is work done in collaboration with Greg Lang, who just started a lab of his own, and with Dan Rice, a graduate student in my group, we evolve these yeast in this sort of batch culture scenario that I told you about yesterday for 1,000 generations. Okay, so our experiment, about half the length of Olivier's experiments, 1,000 generations, haploid asexual budding yeast in rich laboratory media. So this, you know, we specifically didn't want to adapt to something like high temperature where, you know, there's presumably, you know, it's a really novel selection pressure for yeast. This is a sort of more gentle selection pressure. Yeast have been sort of maintained in YPD in the lab for, you know, throughout the history of lab yeast. But as we'll see, they do adapt. And we wanted to sort of just look at the dynamics, right? And just see what we saw, all right? And just a reminder from last time, fitness has this simple definition, which we can measure. It's sort of on average how many individuals survive each bottleneck of a given genotype relative to the other genotypes in the population. All right? So, all right, first of all, if you just look, we did about 600 populations. And if you just look across all the populations, they increase in fitness through the course of 1,000 generations of evolution. Okay? And, you know, that's not really that surprising. These error bars represent the sort of standard deviation between the replicate lines. So this tells you sort of something about how much variation there is. But rough ballpark, they adapt about 1% per 100 generations. They increase in fitness. And that's pretty typical of what we always see in these lab yeast. Okay. And so the thing we decided to do was to sequence through a time course, okay, so that we could see the dynamics. And like I said before, because of library preparation costs at this time, okay, this paper was published about 6 months ago, so I'm not talking about ancient history, but you know, a year ago when we were doing this work, it was, we couldn't sort of prepare libraries that cheaply. And so what we did was we took 40 of these populations, and we picked 12 time points from each of them, roughly every 80 generations or so through the course of this experiment. At these time points indicated by the arrows, we just ground up the whole population and sequenced it, right? And the idea was that, let's say there's some mutation at some time point here that's at 40% in the population. Then what you get is, when you sequence this, you get 40% of the sequencing reads that map to this specific region of the genome. They have this mutant allele at this particular locus, and the other 60% have the ancestral allele at this locus. So we can infer that there's a mutation in the population and that it has a frequency of 40% or whatever it is, right? At the time, we were, this is actually sort of a lot more difficult than it sounds to actually do, because unless you have really a lot of depth, when you're sort of grinding up the whole population, there's a lot of sequencing errors, right? And you're looking for, maybe, as we'll see, anywhere between 5 and 30 or 40 mutations per line out of, you know, 12 megabases of sequence. And so even if your false positive rate is very, very low, you know, you're going to call some errors as genuine mutations, and that can easily swamp your entire data because you've got 12 million base pairs to mess up and only 30 to get right at best, right? And so a grad student in my lab, Dan Rice, figured out how to do this with really sort of moderate sequencing depth. So previous attempts to do this kind of thing had overcome this problem by, you know, having many thousand x depth, which meant that you just couldn't do very much sequencing. We were able to do this with 100x depth, which was what made this study possible, by basically leveraging the time course, okay? And so what we did was, we didn't just look at single time points to look for mutations. We looked at the sort of whole time course jointly. And we said, you know, we filtered out things that had, you know, frequencies that sort of just bounced around in a way characteristic of genetic drift. And only, I mean, not of genetic drift, of sequencing errors. And we only look at mutations that, only look at sites which have frequency which is autocorrelated through time. Okay? And by doing this, we were able to identify basically any mutation that reached at least a frequency of about 10% at any time point through the experiment. And then, given that it had reached that frequency, it sort of went up. We could track it the rest of the time course, above and below that. The little caveat with this is that if a mutation arose right at the end, it's sort of hard to see, right? Because you don't have enough time points. So just keep that in mind. There'll be a little bit of bias from that. All right. This is a sort of a special case of a set of methods that we've now been developing for using some tricks for experimental evolution to do this kind of sequencing. So most of these, I find that actually there's really a huge amount of put it this way. There's a lot of opportunity to do really simple things, to go beyond what most people have been doing with sequencing, because most of these kinds of both the chemistry as well as the bioinformatics of all these things are designed for sequencing humans. And when you're sequencing, relatively few humans, and when you're sequencing microbes, and especially from experimental evolution, you're in a totally different regime, which opens up a lot of opportunities. So the cool thing that we can often do is, we're sequencing many closely related strains, and we're looking for relatively few mutations. And we often know a lot in advance about what the genealogy relating these strains is. I'll show you another example in a little bit of a specific example where, the genealogy has a particular structure. So we can, like I was telling you about with the time course, in any of these kind of situations, sort of construct an error model from each individual sequencing run based on based on all the data, assuming that everything is a sequencing error, and then look for deviations from that error model that obey the known what we know about the genealogy. And you can really get a lot of mileage out of this. Anyway, so we did this across these 40 populations, and this is the sort of zoom out view of what we saw. And I'm going to just zoom in on this to show you. Here are 8 populations. Okay, we did 2 different population size, which I'm just going to call large and small. The small has an effective size of about 10 to the 5, and the large of about 10 to the 6. Okay, so there's about a factor of 10 difference here. The small is not really small in an absolute sense, just relative to the large. Okay. And this is what you see. Each of these lines is an individual mutation that arose spontaneously at some point during the experiment. And we're following its frequency as a function of time from generation 0 to 1,000. Each of these panels is a completely independent line. Okay, I haven't told you anything about what these mutations are yet. I will in a few minutes. But suffice it to say that each of these mutations is a completely independent point mutation. Okay? And we never got, unlike the E. coli lines, with 1 or 2 exceptions, we never got the same nucleotide mutated twice across replicate lines. So each of these is completely unique at the nucleotide level. Okay? I don't know if you can see here, but there are solid lines with circles here. Those are non-synonymous mutations in coding regions. The dotted lines with circles are synonymous mutations in coding regions, like this light blue here. So that's a synonymous mutation. The dotted lines with squares are intergenic regions, and the solid lines with squares are mutations in promoter regions. Okay. So the first thing you can see is that these are definitely rapidly evolving populations in the sense I've been talking about in the last 2 lectures, right? There is a hell of a lot of stuff going on here, right? There are all these different mutations. And they're sort of all competing with each other. So here, for example, we have mutations. This is a population size of 10 to the 5, remember. So these set of 5 mutations increases in frequency, reaches about 70, 80%, and then decides to turn around and go extinct, as it's outcompeted by this competing lineage and this competing lineage here, right? And this is obviously not genetic drift in the traditional sense. I mean, there's way too big fluctuations in frequency to describe by drift. This is just interference. This group got outcompeted by this group. Okay, similar sort of thing happened here. Here we have 3 selective sweeps, but there's lots of other mutations sort of creating additional diversity in these populations. Right, yeah, so this is a good point. So I'll come to this a little bit more detail later on. So because this is just essentially metagenomic sequencing, we don't actually know haplotypes. We don't know the genotypes of the clones. And I initially thought, okay, this is going to be a big problem in interpreting this data. But if you look at this and you sort of apply the rules of logic for a minute, I mean, you can realize it's actually basically almost completely unambiguous what the haplotypes are, right? This is clearly a single haplotype with 5 mutations, right? And there's no other way it could be. Right? Because, I mean, they reach 70% of the population, so they cannot be on different haplotypes, right? They have to be on the same haplotype. Obviously, there may be haplotypes that have 1 or 2 of these mutations and not all of them. But, that can't really be very many because they're all at sort of such similar frequencies and such high frequencies, right? And it's similar sort of things you see in all these lines. You know, some of these low frequency things can't be completely unambiguously resolved to particular haplotypes, but in general, they can all be worked out. Yeah. Okay. So if we just sort of zoom out and look at these things sort of as a whole, what do we see? So first of all, the mutations appear basically linearly through time, right? So you just look, the total number of mutations we've seen increases linearly with time, except this sort of slight dip at the end, which is just due to the ascertainment bias I told you about. And similarly, the number that sort of have either fixed or gone extinct accumulate linearly with time. So what that sort of sum of these two things leads to polymorphism increases and then sort of levels off, right? And you get this sort of steady state polymorphism, okay, after about 600 or so generations, at least for a little while, a few hundred generations. Okay. Now this is, of course, only true on average, right? In individual populations, as you can see, right, there are selective sweeps, there are big extinction events, and so the amount of polymorphism in any individual population is going up and down a lot. And one of the things that really struck us about this, which we still can't explain really satisfactorily, is these big sort of cohorts of mutations, right? So here there's a group of, I don't know, 6 mutations that come up by 200 and some generations and all sweep to fixation together, right? And, you know, here's a group of 5. I think that's 5. Yeah, it looks like 5. Maybe that's even 6. That get outcompeted by this other group of 6, which then gets, you know, helped by this group of 4. You know, you see this kind of stuff all over the place. And we really don't understand how the hell this comes about, right? And so, you know, these multiple mutation dynamics and so on that I told you about predict some degree of clustering in the mutations. I mean, in fact, even for neutral mutations, right? You expect fixation events to be a little bit, to be clustered, right? But, you know, even at 10 or 20% of the population, you see this sort of grouping of mutations that are sort of moving together in frequency. And we cannot find plausible simulation parameter regimes where you get this degree of clustering. So I have no idea why this comes about. There must be some explanation for this that sort of can't be explained by the type of models that I told you about over the past couple of days. Maybe it involves epistasis. Maybe it involves some inhomogeneous mutational process. I don't know. Yeah, they're not clustered at all. So we don't call it clustering because it's not. It's clustering in a temporal sense, but not in a physical sense along. So typically these mutations are on totally different chromosomes. So it's not like a single mutational event or anything like that. I mean, at least not that we know of. Yeah. Right. Right, it can't be explained by that, given what we know about the mutation rates in yeast and so on. And in fact, as we'll see, you know, we have very good evidence that in these clusters, typically many of the mutations will be adaptive. There might be 1 or 2 neutral hitchhikers, but most of them in these clusters are typically adaptive. And yeah, we just can't explain that. Yeah. Well, that certainly is presumably what happened, right? You got one adaptive mutation, then another, then another, then another, very closely together in time. And then that sort of group of adaptive mutations sort of together sort of increases in frequency. But, right. Right, I mean, that would be one hypothesis. But what I'm saying is that, you know, if you assume a homogeneous mutational process, right, you can't generate this degree of anything even remotely close to this degree of clustering like that, okay? I mean, what you can get sometimes is, you know, let me see if I can find an example. I'll show you a better example later, but here's sort of one example. If these were both one mutation, you know, that would sort of make sense. There was this beneficial mutation that occurred, then this beneficial mutation occurred in an individual that had this one, and you know, this sort of group swept first, and this one sort of swept within it, and they both fix at about the same time, right? that sort of thing should be routine, right? Sort of something, a beneficial mutation occurs, it reaches 10, 20% of the population, and then a second mutation occurs in an individual that has the first. But to get this degree of clustering, you'd have to get like the second mutation occurring while the first one was still at frequency 10 to the minus 5. It just doesn't make sense, right? And you, in fact, have to get 5 of them, which is just, yeah. All right. You probably don't need convincing after seeing this, but this is really non-random. It's really significant clustering, right? So if we compute, for instance, just the total number of mutations you saw appear for the first time at any time point, this is what you would expect based on sort of a Poisson distribution in red here. And this is what you actually see. So many more time points where you didn't see anything, and then a big excess of time points where you saw, 7, 8, even 11 mutations appearing at once. Okay. It's just a crude way of quantifying it, what's sort of obvious to the eye. And in fact, right, if you took what sort of Mukund was alluding to, and you sort of said, all right, one mutation of these is a driver, and that's not random. And then all the rest is just random hitchhikers. Right, you'd expect the number of random hitchhikers to be Poisson distributed, right? And it's not, right? So if I just take each of these clusters and I subtract 1 for being the driver, and I look at the distribution of the other numbers, that's also significantly clustered. Okay? Blue is what you see, and the red is what you would expect, all right? I mean, this is, I don't know how seriously you want to take this, but there's plenty of evidence that multiple of these are beneficial. We'll get to that. Right, so what we've seen here is that there are lots of these mutations moving together in clusters. And I mean, the other thing you can sort of get going back to this is, it's not good enough to be a beneficial mutation, right? I mean, none of these mutations, okay? I mean, there are 1 or 2 exceptions, but in general, almost never do you get a single mutation fixing by itself. You just cannot. So, often in like the cancer literature, we talk about like driver and passenger mutations, and like, the assumption is like there's a single driver that sort of drives, that's the whole point, drives the dynamics and fixes, and maybe there's some passengers that hitchhike along. Okay, there is no such thing as a single driver here, okay, with 1 or 2 exceptions. It's sort of like a jointly driven bus here, right? And, it's a group of this group of 6 drivers outcompetes this group of 5 drivers. It's never good enough to just be by yourself, right? And you can see that here in the pattern of repeatability that we see, in contrast to the pattern that Olivier saw in the experiment I told you about a few minutes ago. So these are the mutations, the genes, in which we found mutations in at least 3 replicate populations. Okay, first of all, it's a relatively short list, okay? These are 40 populations. It's, maybe a little more than 1/3 of the population seen in Olivier's experiment. But you see, like, many, many fewer genes mutated in multiple replicate lines than Olivier found. Much less parallelism, okay? And even among the sort of most strongly parallel mutations, this gene IRA1 was mutated in 21 of the 40 replicate lines. And only half the time, a little less than half the time, did that mutation fix. Okay? The remaining time, it sort of goes extinct due to genetic drift, okay? These 2 are seen in about 1/4 of the lines, and similarly, like, they fix a small fraction of the time that they appear. So each of these mutations is appearing way more than you would expect by chance. You'd essentially never expect, given the number of genes in yeast and so on, you'd never expect to see any gene mutated 3 times or more by chance. So these are presumably, strongly beneficial mutations. Okay? And in fact, DNDS among this group, I think there's 1 synonymous mutation among this entire group, right? So it suggests these are really beneficial mutations, but yet, even these mutations only fix a relatively small fraction of the time. So it's just reinforcing that it's not enough to be beneficial just by yourself, right? Yeah. Right. Yeah. There's a stronger directional selection in the E. coli case. So presumably there are bigger differences in fitness. Okay. The population genetic parameters are not quite the same, right? The mutation rates in yeast are higher. right? So a per base pair mutation rate in yeast is maybe 10-fold, maybe 5-fold. I mean, it's controversial, right? But it's a few-fold higher than in E. coli, and the genome is twice as big. So you have a mutation rate, genome-wide mutation rate, maybe a few times higher, maybe as much as 10 times higher in the yeast than in the E. coli. The population sizes are ballpark similar. Yeah, I mean, but the structure of selection is presumably quite different, all right? And this is the kind of thing that, I mean, I'm just sort of raising the question, right? I don't have an answer here, but this is the sort of thing we would like to be able to explain with the kind of theory that I told you about over the past couple of days, okay? And it's a sort of thing, the theory is sort of, I mean, it's clearly relevant, but it's not directly touching these experiments yet. And that's sort of where I think we need to go, right? Both with the experiments and with the theory. All right. And we're sort of, yeah. I mean, no, not much. So we are in the, I'll tell you about, we are sort of in the process of really systematically dissecting the fitness landscape in this case, okay, via various kinds of experiments, which I'm going to tell you about in the next 45 minutes. You know, the E. coli, we know less about the fitness landscape, actually, and it's harder to dissect for reasons which I'll tell you about. But, I mean, yeah, I mean, we would like to be able to say from these kinds of differences, what must be the differences in the selective pressures, and then be able to verify that in some way, right? And that's sort of the goal of all this stuff, but we're, you know, not there yet, but no, that's sort of, we haven't done it, and that's the question which I want to leave you with, I guess. Yeah. No. So, I mean, one of the things we have sort of thought about this clustering is there could be sort of very short time scale physiological fluctuations in the mutation rate. That's one potential explanation for this stuff. You know, there certainly is some of that. For instance, these things are growing in batch culture. So the environmental conditions are changing, right? And you know, every day over 10 generation timescales, the conditions get bad, they get good, they get bad, etc. And so maybe this leads to fluctuations in mutation rate. The question is, how much of a fluctuation in mutation rate would you need to explain this kind of data? And you would need actually quite an enormous amount. And so we can sort of, we can try to measure that. I mean, I think that would be sort of cool just from a molecular biology point of view as well, if there were really that big fluctuations in mutation rate. There are other potential explanations, though, and yeah. No. We don't have enough time points yet to really test that. We're working on more, but no, from what we have, there's, right, at least as far as we can tell, there's no change, okay? But yeah, I wouldn't say for sure if we were to go longer, we wouldn't get some change. Yeah. All right. These are what the mutations look like if you care, but I'm going to skip that. So, all right. We can sort of across all the populations say something about what mutations we observe and what mutations fix. And I just think this is kind of interesting to look at. So we have about 1,000 mutations total, and about a quarter of them fix over the whole experiment, right? These are the numbers of intergenic, synonymous, and non-synonymous mutations. The first thing I just want to look at is the synonymous versus the non-synonymous, right? We see about 100 synonymous, about 600 non-synonymous, and they fix at almost identical rates. Now, of course, this is a little biased. We're only looking at mutations that reach at least 10% at some point or another, right? But conditional on reaching at least 10% frequency in the population, you know, there's almost no difference in fixation rate between synonymous and non-synonymous mutations. And this is sort of subdividing the non-synonymous mutations into those that are sort of one-offs, those that were hit in exactly 2 replicate lines, and these are the sort of multi-hit mutations. And even those, you see, only fix a little bit more than 1/3 of the time. Okay? And you see sort of if we break down the small and large populations separately, you see pretty similar things between the small and the large populations, although the fixation probabilities of everything, conditional on reaching 10%, remember, are much greater in the small populations than the large ones. It kind of makes sense. I'll describe to you in a bit more detail in a minute. But I mean, sort of intuitively, given that you've reached 10% in the small populations, given that you've occurred and reached that frequency, there's sort of less interference in the small populations. And so there's less sort of stuff to jump beyond you and and screw you up, and so you fix with greater probability. But similar patterns hold with the synonymous versus the non-synonymous and so on, right? And so we can sort of interpret this. Well, I'm going to fall off this stage. We can sort of interpret this intuitively in terms of the theory that I told you about. Here, let me just show both of these. So each of these trajectories, I'm just drawing a sort of schematic trajectory here. We can extract various information from it. So when did the mutation first appear? How high did it get? How quickly did it initially spread? And I want to focus on this parameter here. How quickly did it initially spread? So on a log scale, you can just sort of fit a line to the initial increase in frequency, and I'll call that S up, okay? And so we can interpret that in terms of the theory that I told you about in the past few days. So at a given moment, right, there's some distribution of fitness within the population. Okay, I'm drawing it as this sort of light blue line, this sort of Gaussian distribution here. Okay, you know, it'll fluctuate around, of course, but on average, it should have this sort of shape. Okay, and a mutation occurs in some genetic background, right? And it lands at some landing fitness, right? And that landing fitness is a sum of its own individual effect plus the genetic background on which it occurred, right? Like I was telling you about the past couple of days. And so then its landing fitness is described by some sort of total fitness, which is the sum of these two. And then, you know, once it lands somewhere, its future dynamics are determined just by its landing fitness, right? Not by anything else, right? Like I was telling you about. And, you know, in particular, this initial increase in fitness, okay, is a measure of that landing fitness, right? Because that initial increase in frequency is determined by how much more fit it is than the average of the population, right? Now, we're not quite measuring that because we only see it when it gets to about, you know, 10% of the population. But, you know, we're seeing it before it gets at least too frequent that it's significantly affecting the mean fitness itself, right? So we see something, you know, at least ballpark close to this landing fitness. And then what would happen, of course, the mean fitness is sort of catching up to you. And if nothing else were to happen, this guy would go extinct. Of course, you know, additional mutations could happen in that background. It could sort of surf ahead and it could fix, right? So this would be these two sort of parts of this W of X, right, that I talked about. Whether it fixes or doesn't is determined by then what happens, whether the rest catches up or whether it sort of manages to stay ahead, okay? But a prediction of this sort of picture would be that the fixation probability increases with the landing fitness, okay, and does not depend on anything else, right? So given the landing fitness, given this S up, it shouldn't matter whether the mutation is synonymous or non-synonymous or in a multi-hit gene or in a one-off gene or whatever. It should only depend on this S up. And in fact, that's what we see. So if you look at all these mutations, and we look at this initial S up, that's what I'm plotting here, that initial sort of landing fitness, and the fixation probability increases with the landing fitness, okay? But it's basically indistinguishable whether you're an intergenic, synonymous, or non-synonymous mutation, whether you are going to fix, given that you land somewhere. Now, there's not as good statistics as we would like to have here, but at least as far as we can tell, this picture seems to generally hold, right? And it's suggesting a good mutation on a bad background is as likely to fix as a bad mutation on a good background, right? It's just the landing fitness that matters, right? Okay, so at least that much is consistent with the sort of theory we talked about. We can also sort of separate small and large populations. So this is the same sort of figure. Now I'm dividing up small and large populations. And this here shows that given that you land a certain amount above the mean, if you land a certain amount above the mean in a small population, you're more likely to fix than if you landed that same amount above the mean in a large population, right? And that makes sense, right? There's less variation in fitness in the small than the large populations, right? Those traveling waves are narrower in the small than in the large populations. And so what that means is that given that you land a certain distance further out, you're sort of further ahead of the wave in a small population than a large population, so more likely to fix. Of course, at the same time, you're more likely to land further out in a large population because there's more genetic backgrounds that can sort of get you out there in a large population. So this sort of doesn't show that, but in a large population, you're more likely to land out here than you are in a small population. But given that you land at that far ahead, the small population is better. Okay? So this is sort of leads to a little bit of a trade-off in the repeatability, right? There are less mutations that occur in the small population. So the large populations get the sort of best mutations more frequently just because there are more mutations happening. So they're more likely to sort of sample the full set of mutations that you could get in the large than the small populations. But given that you happen, right, you're more likely to fix in the small population than the large. And so that sort of slightly sort of counteracts the effect of the large populations in finding the best mutations in the first place. Okay. I'm just going to say a few more things about these kinds of experiments. So, you know, as I was mentioning in response to Mlchael's question earlier, if you just apply the rules of logic, okay, this is one particular example of a population trajectories, and if you just apply the rules of logic, it's sort of fairly easy to figure out what the haplotypes are. And you can just do clustering and apply these rules, and then you can get from there to this Muller diagram. Okay. So this is just an example which, you know, just sort of illustrates all of these kinds of things that happen, right? So in this particular population, you got this mutation in SBC3, okay? Non-synonymous mutation. This was the only population in which this mutation occurred, a mutation in this gene occurred. Similarly, in a competing lineage, you got this mutation in this putative whorf here, okay? When you see these long names, it means nobody knows what the hell this does, or, you know, anyway. You get this. Again, this was the only population where this mutation occurred, but it seems to be beneficial. It's increasing in frequency, okay? That's this one and this one here, this one and this one here, okay? They increase, and then this group of 6 mutations appears, okay? It includes mutations in ROT2 and WE2, which occur in 11 and 7 of these populations respectively, okay? Plus some of these, this guy appears in only one other population, and then there's this other stuff, okay? And this group of 6 mutations is sort of out-competing everything else, all right? But then, right, this set of 3 mutations, including UR1, which is one of the sort of most commonly hit mutations in the whole experiment, and DOT6, which is also present in one other population. This group of 3 mutations occurs, and it, you know, comes about. And then this mutation in WE2, this green one here, occurs. And this now group of what's now 6 mutations is now out-competing this group of 5 mutations. Sorry, this other group of 6 mutations. You notice there are 2 WE2s here. There are 2 independent mutations in WE2, different at the nucleotide level, but in the same gene. Okay. And so this group of 6 is out-competing now this group as well as this single guy who's now completely toast. And now this group of an additional 3 mutations starting to appear in this subclone. And it's going to drive it to higher fitness. Sterile 4, this is a mutation we know to be beneficial from other work. HXT14, this is the only population in which we saw this mutation, but this is a hexose transporter, almost certainly beneficial in this kind of regime. It sort of transports glucose. All right? It's just kind of funky. It just, I mean, illustrates all these kinds of effects that I've been talking about over the past few days. Yeah. Yeah. No. There's no time dependence to anything, right, in terms of when mutations appear, as far as we can tell. I mean, it's only 1,000 generations, but yeah. All right. So in the last year or so, I guess, I don't know if this is right. Maybe it was just, maybe this might even be 2014. Just in the past couple months, Dan Kwiatek and Gavin Sherlock, who we talked to a lot about while we were doing this stuff, had a paper on a similar sort of experiment in yeast, in this case in chemostats, at a population size maybe 3 or 4 orders of magnitude larger, population size between 10 to the 9 and 10 to the 10 here. Okay? And these are the mutations that they see in the Muller diagrams. They see they only had 3 populations, okay, but much larger population sizes and a little bit less time scales, about 450 generations. And you see sort of vaguely similar sorts of things going on in these populations. And in fact, you know, there's sort of strongly overlapping mutations that you see. Some of the same ones again, some different things, but some of the same mutations you see in their experiment as you saw in ours. Okay. This is, you know, much, much larger populations, though. There's not really enough replicate populations here to draw any real comparisons with our data, but just interesting to see roughly the same set of things. Now, to get to one of Kavita's questions earlier, one of the things which we're actively doing right now, and we're actually, yeah, it's a real pain in the ass, but what we're doing is we are trying to map the fitness effects of all these mutations. I've sort of told you sort of about evidence that these mutations are beneficial, sort of indirect evidence, but we can actually, one of the nice things about yeast is we can do this directly. We, in this particular figure that I'm showing you, we took a bunch of clones of yeast from this time point of our experiment. And we also isolated a few ancestral clones. And we measured their fitness. So these are fitness of 7 evolved clones from this populations, ranging in fitness from 3.5 to just over 5%. And we also took the ancestor and measured its fitness relative to the reference strain that we're using here. And so that's what that is. And then we crossed the evolved strain to the ancestor. And you just mate them, and you let them sporulate, and you isolate a bunch of spores, and we measure the fitness of each of those spores, and we genotype each of those spores for each of these mutations that we know is present in the evolved population. And this is what you see, the relationship between the genotype of the spores and their fitness. And so from this you can see, right, so the individuals that have none of these mutations are as fit as the ancestors. The individuals that have all of them are actually a little more fit than the evolved lines. Okay, and then if you have the gas one and the sterile 12 or the sterile 12 mutation, you have a fitness advantage of about 3%. The ELO mutation appears to be a neutral hitchhiker. That's this blue one here. Okay. And then these double mutation of the gas and the sterile 12 have this fitness advantage. It's there's maybe some slight negative epistatic interaction between these two, but the double mutation is more fit than the single. And the triple is the same because this ELO one appears to be neutral on all of these genetic backgrounds, right? So this population we picked for this purpose as a proof of principle because it's the simplest one that we saw. We're now in the process of trying to do this with sort of all these populations and just map all these fitness effects. Unfortunately, it's a sort of massive pain in the ass because many of these evolved lines become sterile. When I say sterile 12, that means that this line is sterile. Which obviously complicates this process. You have to complement the sterility with a plasmid and then boot out the plasmid, and it's a pain. But we are in the process of doing this. So eventually we'll know. All right, I'm going to just skip this because. So what I've told you about is all of this sort of population sequencing data, and if we want to connect it directly with the theory I told you about. The past few days, we need to sort of translate from what I told you about genealogies to the trajectories of these mutations, okay, over time, which is something we've worked on doing, and I just skipped over like how we go about doing that, because nowadays we can actually, now that we've solved this library preparation problem, actually just sequence a bunch of clones from these populations and also just directly see the genealogies and compare them to theoretical predictions. And we just got this data like a few days ago. I was just looking at it in the back of the room. So I just thought I'd show you. This is the first example of that. So we sequenced from 4 populations, just as a start. From 4 populations, we sequenced 12 clones at each of 2 time points. So this is just looking at the tree made by these. These ones shown in red are from a late time point. This is from an earlier time point, and this is actually, in this population, we also sequence a couple of clones from an even earlier time point. And you just see what the tree looks like. I don't know what to make of this yet, but you can see all the mutations and so on and the branching structure of the tree. It's clearly highly non-neutral, which is not surprising. But we'd like to sort of interpret this. It's also maybe interesting to look at this on the sort of Muller diagram. This is the Muller diagram relevant for this particular population. Here now each of these clones is put according to, that we sequence is put according to when it was sampled from, and you can see the genealogy that you get. Okay, and you can see these sort of signatures of of highly unbalanced trees and multiple sort of branching events, but not a single selective sweep. So there's this theory on these multiple merger coalescence, which this does appear to sort of look vaguely like. All right. Here's another example. This is population 2 that we saw, okay? Looks quite different, but also a lot of these sort of multiple mergers. So those first two were small populations. This is a large one of the large populations. You know, you see, again, this kind of highly unbalanced tree. Here's the other large population that we sequenced. Looks kind of weird to me. All right, just to sort of see, these are the 4. Okay, and one of the kind of weird things about this to me, these are the 2 small populations, these are the 2 large populations. If I didn't tell you that, it would have been probably pretty hard for you to tell the difference, right? There's a factor of 10 difference in population size here. You know, the neutral results that we saw, of course, which would suggest enormously different genealogies in these 2 populations. And clearly, because of this interference that's going on, I mean, you couldn't tell from the genealogy, at least not just at a glance, that which of these was the large and which was the small populations. This is looking at that same data just at the trees. These are, again, the small populations. These are the large populations here. So anyway, if anybody has any ideas about what we should do with this data, I just sort of wanted to do a little pilot to just look at what the hell this looked like. I have no idea what I'm going to do with it yet. All right. So the last sort of thing I want to tell you about is just some experiments to look at this landscape more directly, right? So we saw in the experiment that I was just telling you about big differences from Olivier Tenelian's experiment, sort of qualitative differences in the degree of repeatability, and somehow that must reflect differences either in the dynamics or in the possibilities, or more likely both, right? So there's another class of experiments that we can use to sort of map aspects of these possibilities a bit more directly, okay? So I'm going to sort of go quickly through the introduction, but you know, this sort of set of possibilities can be sort of characterized in various ways, right? What we really care about is sort of what's the landscape on which, you know, what's the sort of fitness landscape, or however you want to define that. What are the mutations that can occur? What are their mutation rates? What are their fitness effects? What are their interactions? And people try to map this with, you know, some Fisher's model, or with fitness landscapes, or, you know, there are folks who try to sort of take these sort of maps of metabolism and sort of directly, you know, think about what the landscape this might imply. What I'm going to talk about is these sort of local descriptions. You know, we have some distribution of fitness effects of mutations. So you get a mutation. What is its fitness effect? That distribution has some shape, whatever the hell it looks like. And that's something that a lot of people have worked on just directly measuring, by just making a whole bunch of mutations and measuring their fitness effects. And so there's a lot of, you know, attempts to experimentally measure this. They're sort of fairly crude and coarse descriptions, but you can sort of try to measure this directly. There's a lot of work on that. What I want to talk about is a sort of slightly less local thing, sort of sets of epistatic interactions between mutations. Because over the past few years, you know, one signature of this was Olivier's experiment, but even before that, there's sort of been a lot of sort of talk in the literature that epistatic interactions, strong epistasis between beneficial or potentially beneficial mutations, is really important in understanding these experiments. So, you know, this is one of the sort of first studies of this sort, where Dan Weinreich, I think he was in Dan Hartle's lab at the time, looked at mutations involved in the evolution of penicillin resistance. And, you know, he looked at mutations in beta-lactamase, which is a main gene involved. There were 5 mutations in his experiment that went from some wild-type state to some highly resistant state. And, you know, he made all possible combinations of these mutations and found that many of these pathways, many of the orders of mutations you could get, involved really strongly deleterious mutations. So the idea behind this is there's sort of strong sign epistasis. There's only a few specific orders in which you can get these mutations, in which each step was sort of towards increasing resistance, right? And the idea, sort of intuitive conclusion of this work, was that you need to have a specific order of the mutations, right? That epistasis is really significantly constraining how the population can evolve. More recently, Chris Marks, so this is like, as a function of the background fitness, the fitness of individual mutations can have some sort of highly idiosyncratic effect. On some genetic backgrounds, some of these mutations are highly beneficial. On other backgrounds, the same mutations are deleterious, okay? Or certainly not beneficial, okay? So there were a couple of papers, one from Tim Cooper's lab, one from Chris Marks's lab, in Science a few years ago now, showing that if you take mutations that occurred in a sort of single population as it evolved, in this case from Rich Lenski's long-term evolution line, and in the case from Chris Marks's lab in Methylobacterium from one of Chris's evolution experiments, and you make all possible combinations, they found a sort of more consistent pattern. I'm going to just skip a little bit. This is Chris Marks's figure. What he found is that if you take each of these mutations and you look at its fitness effect as a function of the background, as a function of the fitness of the background in which it occurs, you get this nice negative relationship of diminishing returns. So fitness effects of individual mutations do depend on the genetic background, but in this kind of more systematic way. Okay. But all of this, these 3 studies that I've told you about were among, 5 mutations, 4 or 5 mutations that occurred along a single evolutionary trajectory. And so, you expect these mutations to play nicely together, right? Because they occur in the same population, so, at some level they have to be compatible. I mean, there could be orders that don't work, like in Dan Weinreich's experiment, but in general, they're you're constraining them to sort of work together to some degree. And so the last thing I want to tell you about, this, could obviously explain these slowdowns in adaptation. The last thing I want to tell you about is experiments that we did very recently, and this is not published yet, but work that we did recently to try to understand more generally how does epistasis constrain the way that the populations evolve in the future. And if you think about it, in order for epistasis to matter in terms of how the population adapts, it's not enough for an individual mutation to have epistasis with lots of other individual mutations. What you need is for an individual mutation to actually change the DFE going forward from there, right? An individual mutation has to change the sort of average fitness of, it has to make beneficial mutations, let's say, much less likely or have lower fitness effects in general in order for it to slow down and constrain adaptation. Or for it to open up new possibilities, it has to be the case that you get one mutation and then the whole DFE has to get, the whole distribution of fitness effects has to get better. Okay? And so we set up this experiment to try to get at this question. All right? So we took this ancestral clone of yeast, okay, which we're going to call divanc, and we let it diversify in about 500 independent replicates. I'm just showing a few here for simplicity. And from that set of 500 replicates, we picked one clone each, okay, and we measured the fitness, and we picked a subset of 64 clones that spanned a range of fitness, okay, and we call these the founding genotypes. This was about 250 generations of diversification. So each of these clones differs from each other by 500 generations of sort of independent evolution, right? And there's about, on each of these branches, there's about 2 to 5 mutations. So these mutations differ, these clones differ by 4 to 8 mutations, something like that, okay, these founding genotypes. Then we took each of these 64 founding genotypes, chosen to span a range of fitness, and evolved them in 10 replicate populations each, 10 independent replicate populations, right? And so the idea here, I'm just showing 3 for simplicity, but just so I could fit it on the graph, there are 10 independent here. And what we want to do is look basically, we want to ask the following question. If the 10 replicates that are founded from, the same founding genotype, they're not all going to be identical, right? And that's a reflection of just sort of the inherent randomness of the evolution along these branches. Okay? That has nothing to do with the founding genotype because these guys all have the same founding genotype. But if clones descended from founding genotype 1 differ from clones descended from founding genotype 2, 3, up to 64, in a way above and beyond the variation within these groups, then that's an indication of some effect of this founding genotype, namely effects of mutations that occurred in these branches in sort of setting what the possibilities were going forward, right? So that's what we wanted to investigate, okay? All right. So we did this, all right? And I should just say, so we did this. This was 250 generations, because we wanted to do like sort of, you know, short time scales, a few mutations that you would see in an evolution experiment. Does it sort of affect what happens going forward? And then, you know, this is 250 and then 500 generations. So we did 500 generations on here, and we also took a time point at half that time. What you see, okay, this is the distribution of fitnesses of these founding genotypes in light blue. And as time goes on in the adaptation, they get fitter. Okay, that's not surprising. And this variance gets smaller, so they get sort of closer together, okay, across all the population. So they sort of adapt and they converge. And the first thing, okay, ignore these insets for the moment, all right? If we just look at if we sort of do an ANOVA to partition the variance in final fitness, okay, what we see is a real strong effect of the founder genotype on how you adapt going forward. Sorry, this is not final fitness, because obviously final fitness is going to be related to the founder genotype because they have different, this is how much your fitness increases over the 500 generations of adaptation. That fitness increment, that is strongly influenced by the founder, right? So about, you know, some 15 to 20% after 250 and 500 generations of this is just pure measurement noise. These are errors in our fitness assays. About, you know, half, and then later about a third of the variance is explained by just the stochasticity, differences between replicates. But the founder genotype actually does have a real strong effect. So these different founders have big differences in their how they adapt going forward. All right. However, there really is a strong convergent evolution for fitness. So if we just look at all the data, right? This is the initial fitness of a clone, okay, one of those founders, and this is a final fitness of the population. So there's 640 points on here, one for each of these populations. Each has its sort of initial fitness and final fitness in this sort of green after 250, and in this yellow after 500 generations of adaptation, right? And you can see that these things are sort of converging in fitness over time, right? This black line is what would happen if you didn't evolve at all. Right? So they all are sort of adapting, but the guys that started at lower fitness are adapting faster than the guys that are starting at higher fitness. Okay. There's a lot of spread here because of this sort of inherent stochasticity in the process, as well as measurement error, of course, but mostly just randomness in the process itself. But we can average out that spread because of the design. So we can take all 10 clones founded from a given founder and average their final fitness. Okay, and that's sort of averaging over the stochasticity between the lines founded from the same founder and asking on average how fast do the lines founded by this guy adapt. Okay, and so we do that, and this is what we see, a much, much cleaner picture. Okay, so this, for example, is a, you know, given low fitness founder, and you know, it was initially at this fitness, it adapted, it increased in fitness by the difference here, okay, to get to this final fitness. And these error bars are the you know, standard error of the across these different replicate populations. Okay? And so what you can see is that almost all of the effective founder genotype is explained by the fitness of the founder, right? So these founders got to this initial relative fitness in totally different ways. They have completely different mutations. I'll tell you a bit about what the mutations are in a few minutes because we've sequenced to see what they are. And they're in, you know, totally different biological processes, no real rhyme or reason to it, right? They got to two founders that are at this fitness are totally different genetically, yet they adapt at exactly the same rate and different than founders that were at this fitness or this fitness. Okay? And so, you know, if you partition this further, that's what these subgraphs are. Almost all of this effect of the founder genotype is explained by the founder fitness, and only a small sliver is explained by aspects of the founder genotype other than its fitness, right? These are statistically significant, and we've actually further confirmed them, right? There are differences, there are cases where there are two founders of the same fitness that adapt, and, you know, one adapts much faster than the other, okay? And, you know, that we've done, we just repeated the experiment, you know, in many more replicates on pairs of those to just confirm that's the case. But that's pretty rare. Most of the time, everything is explained by just this founder fitness. All right. So we wanted to look at like why is this, right? Okay, so one obvious example, one obvious reason for this could just be, all right, there are 8 mutations you can get. You know, kind of like Olivier's picture, right? There are 8 maybe pathways that you can mutate, right? And you get mutations in them, and then you're done, and you've adapted, and this is a sort of fitness peak over here. Once you get to this fitness, you can't really get any fitter, and then you're done. And you know, these guys adapt faster just because those founders haven't got any of those mutations yet, and these guys adapt slow because they already got all those mutations. There's nothing left, right? Yeah. Sorry? Yeah, this is also YPD. This is the same sort of scenario that I was telling you about in the previous experiment. Well, it's competitive fitness. Yeah. Yeah. No. Yeah, it's almost changed. I mean, maybe a few %, but. To the extent that that would happen, you would expect the opposite relationship, where the more fit populations would have a higher density and adapt faster. But those differences in density are really tiny. Yeah. Yeah, so we don't really know yet. Right now, I mean, I'm not going to have time to tell you about it, but we're doing these experiments also in a whole panel of other environments and with a whole bunch of varying some of these parameters to see how it's different. Okay. Particularly, we're looking at the environmental stress. We have a panel now of 13 environments, and I'm not going to have time to tell you about it, and we don't really have a clean answer yet. But yeah.
Is there some way in which you could turn up or turn down the mutation?
Yes, you could use mutator things and so on. And the problem with this sort of stuff is that, you know, there are a lot of things we can do. We're working on these things, but we are not playing with mutation rate yet, but we could. Yeah, there's only, I mean, even with the robots, there's just so much we can handle. Okay. All right. So we took 13 of these founding genotypes, and we sequenced 130 lines. When we started this, we hadn't really completely worked out these new library preparation methods. Now we could do more if we wanted, but. Okay, so first of all, there is significant convergence, okay, like in our earlier yeast experiments. But just to compare with Olivier's, right, this is the same rarefaction curve in light blue as this light blue over here, roughly the same number of lines. It just looks totally different, right? There's much less convergence in these populations than there is in Olivier's. I don't know why. Okay. These are the mutations we see, okay? We see, you know, you can classify them into all of these categories. Right? And so the first thing we wanted to look at is just, okay, how many mutations did each of these lines get? All right? So we're looking at these sort of putatively functional mutations, basically, you know, the non-synonymous and promoter, frameshift, these sort of orange mutations here. And what you see is, okay, these are the different founders that we sequenced, and these are their fitnesses. So, you know, spanning a big range. Okay? And you can see there's really no correlation, there's no tendency for, I mean, if anything, these sort of high fitness founders on average get a few more mutations than the low fitness ones, not the other way around, right? So it does not appear to be the case that these guys have just sort of, you know, they already had all the mutations and these guys didn't have any of them, right? And so these guys just adapted faster because they got more mutations, right? And you can sort of, you know, look at this in many different ways. You could, instead of putatively functional, you could look at all the mutations that occurred in genes that were hit in more than one replicate line or whatever. No matter how we look at this, there's just no correlation, right? Does not appear to be any of this tendency like this that you would expect if it was just a simple running out of mutations. Okay. Another thing we looked at was, you know, is there an excess of what we call parallelism over convergence? Okay. By that I mean, okay, let me just explain this. Okay. If we take the 10 lines descended from this particular founder genotype and ask, you know, among all of the genes that were mutated in at least 2 lines, okay, how many do they share with each of the other clones that we sequenced, okay? And so we average that then among all the clones that are descended from, you know, sorry, this should be, oh yeah, it's drawn this way. So here, this is from this founder and this founder. This square is saying how many do they share with the other clones descended from the same founder. This box here is saying how many does this clones descended from this founder share with clones descended from this founder, and so on, right? And so this here on the top left, this is the number of shared genes. Here we sort of did a coarser level of aggregation by grouping genes into what are called gene ontology categories. There's sort of coarser groupings of genes saying, you know, genes with similar functions, kind of along the lines that Olivier's experiment group genes into functional categories, okay? And I mean, the thing that I want you to take home from this is that there is really no structure that we can see here. If it were the case that there was more parallelism than convergence, in other words, lines descended from the same founder tended to be more similar than lines descended from different founders at the genotypic level, okay, then you, as you would expect under this running out of mutations hypothesis, then you would expect these sort of diagonal elements to be darker than the rest of the graph, and you don't see that. Furthermore, these lines are organized in order from lowest initial fitness to highest, and you see no sort of further structure either, where high fitness guys tend to evolve more similar to each other than low fitness guys. There just appears to be no structure whatsoever, right? So, you know, at the genotype level, it just, despite the effect that the founder genotype does really matter in terms of how fast you adapt, there's no signature of that in terms of functional categories of mutations or genes in which mutations occurred. All right? And this is just another way of looking at that. Okay, looking at these. These are the founders. These are the mutations in the founders. Okay, the non-synonymous mutations in coding regions in those founders. You can see what they are. And these are the mutations that they got. I mean, obviously they got also a bunch of mutations each that was unique to individual clones, but these are the mutations they got in genes that were mutated in 3 or more replicate lines, and there's just no real sort of rhyme or reason to this, right? It's not the case that individual founders got specific mutations that other founders did not get, okay? So the question is then, where the hell does this sort of convergence come from? Where does this effective founder fitness come from? And so, you know, one hypothesis is that it's sort of diminishing returns along the lines found by the Marx group and the Cooper group. And there's sort of two ways that could be, right? What needs to be the case to get this sort of, to go back, to get this result, is it needs to be the case that on average, mutations have a lower fitness effect in high fitness backgrounds than in low fitness backgrounds. Okay? That sort of the average mutation has a smaller fitness effect in these backgrounds on the right than the backgrounds on the left. Okay? This doesn't have to be the case on the level of individual mutations, right? And in fact, I mean, it seemed to me, at least, thinking about this, weird that it would, right? Like, it could just be the case that there are less beneficial mutations available to the high fitness than to the low fitness background. And so that would be sort of on average diminishing returns, but not at the level of sort of individual pairs of mutations, right? You know, it could be the case that just, you know, but it could be the case that just, you know, every single beneficial mutation is still beneficial in all of these backgrounds. The set of available mutations was the same, but just consistently, all the mutations had a lower fitness on the high fitness backgrounds than the low fitness backgrounds, which we would call a sort of a more universal diminishing returns at the level of individual mutations. So either of these things is possible. And so what we did was we actually took individual of these mutations. We actually took 3 of these mutations that were commonly mutated across all these lines. Okay, these are 3 of the sort of more common mutations here. And in fact, we picked the ones that showed the strongest signature of parallel evolution over convergent evolution. So these are the ones, if you were going to get a sort of idiosyncratic effect between different genotypes, these are the ones where you would expect to find it. Okay? And we made these mutations. Actually, these mutations, obviously there were different mutations in different lines, but they all appeared to be sort of loss of function, you know, premature stop codons and things like that. So we just made these deletions. And we made them in each of the various founder backgrounds, as well as a few other strains, just to test. We also took a control, an HO delete. Okay? These lines that we started with have a frame shift in HO, so this should be a sort of neutral control. And we made all these deletions and all these backgrounds, and here's what you see. Okay, there's a nice, clean relationship. These backgrounds have all kinds of different mutations, yet the fitness of the background and not the individual genotype seems to determine what the fitness effect of each of these mutations is. So, you know, as weird as it may seem, sort of when I explain it, there does appear to be this sort of universal diminishing returns epistasis, where the fitness effect of a mutation doesn't depend on, you know, all the details of the genotype of this background, but only on the fitness. Even though, you know, these strains got to these fitnesses in very different ways, you know, with different biological processes, you get all these different, nevertheless, it's just the fitness that seems to matter. Yeah. Yeah. No. Well, I mean, we just had some experience from previous work that there would be a few mutations. We didn't want there to be, you know, huge numbers of mutations because we wanted to look at this kind of path dependence of evolution on the sort of timescales of these kinds of evolution experiments, right? Now, another thing that would be interesting to do, for instance, is to take sort of two divergent wild strains of yeast and cross them and get a bunch of, you know, strains that differ by tens of thousands or hundreds of thousands of mutations and ask this same kind of question. And that's one of the things on our list. And then, you know, maybe over those much longer evolutionary timescales, there are different sorts of results. And that is obviously relevant if you're thinking about outcrossing populations in nature and so on. But for this, we wanted to look at things sort of relevant for the lab experiment. So that's why we chose this. Yeah. That's also on the list, but not yet. Yeah. I mean, see, to some extent, what this implies to me is that there is just one phenotype, right? And it seems kind of weird, right? Because, like, just like a few words, I'm going to go slightly over, sorry, but just a few words about, like, you know, we sort of feel like the main selective pressure in these experiments is that the yeast have to learn to trust us. So, you know, the yeast, right, they have sort of two physiological modes. If you had to divide it sort of coarsely, I mean, obviously there's a lot of substructure, but if you had to divide coarsely, there are two physiological modes. One of my postdoc mentors, David Botstein, calls it shields up and shields down. When things get stressful, shields up, growth rate down, you know, batten down the hatches. And, you know, when things are great, you know, shields down and, you know, bring everything into the cell and grow, grow, grow. And that's risky. And, like, you know, if you take yeast in nature, like, they're really, like, under stress a lot of the time, right? So shields up is, like, the dominant mode. And, you know, it has paid yeast to be pessimistic, right, in the past. But now they're in an environment where, like, the graduate student is going to show up every 24 hours, right? Or I'll be pissed. And so, you know, the yeast learn to sort of trust the graduate student. They know conditions are getting bad, but you know, the graduate student is going to come along, and so they can, you know, they can try to eke out that additional cell division and not go to shields up mode quite yet, because, you know, the conditions are going to get good again soon. And there are a lot of mutations that are sort of consistent with that. So, like, the IRA1 and 2 are negative regulators of RAS. RAS is like go, go, go. And so you knock out the negative regulation of RAS, and, you know, you're just going to sort of tend to try to divide, divide, divide. But there's a lot of stuff that isn't, that clearly, like, has nothing to do with that, at least as far as we can tell. There's stuff involved in membrane structure. There's the sterile mutations, which is just basically turning off stuff that sort of is like metabolic load due to production of unnecessary proteins. Yeah, I mean, there's all kinds of stuff. There's glucose transporters. I mean, there's stuff that does not appear to have anything to do with one another, with each other. But yet, yeah, somehow it appears that there's like one phenotype that, or everything is sort of coupled together through one thing. I don't know. Yeah. Yes. Underway. Yeah. That's the other thing you could do, right? Take these different founder genotypes, mutagenize them, ask, do they, yeah. Yep. Yeah, so I mean, there's two things that we're doing. One is just, right, you can generate these founder genotypes in various ways, right? You could generate them by mutagenesis and look at that, okay? I mean, the other thing you could do is take these evolved lines and mutagenize them and now ask, you know, if you go down in fitness by a given amount, even though you've just adapted in this environment, still does the fitness predicted in the same way, right? Another thing you could do is actually ask, rather than allowing these founders to adapt, you could let them decline in fitness, right? And ask if there's an effective founder genotype on how fast you decline in fitness at either a small population size or under mutagenesis itself. So these are all things that we are doing right now. Okay, I should stop there. There's various other stuff I could tell you about, but I'm going to stop. And you know, hopefully this just gives you a sense of all the sorts of things that you can do with experimental evolution and the kinds of you know, some of these have close connections to theory, like I was telling you about earlier, and these kinds of latter types of experiments are more about sort of parameterizing, you know, what sorts of landscapes are realistic and what sorts of issues do we have to consider for future theory. I just want to end by acknowledging people in my group who have done a lot of the experiments that I told you about from our lab, including particularly Elizabeth and Sergey, who have done this sort of a diversification experiment that I told you about, and Dan and my collaborator Greg Lang, who were doing that first experiment that I was telling you about. And a lot of the theory that I was telling you about, you know, I told you about sort of general work in the field, but work on that area that's ongoing is being done primarily in my group by these 4 people, and of course by a lot of other groups as well. All right, stop there. Thanks.